This protocol describes how to induce liver vesicular steatosis in differentiated HepaRG cells and methods for detection and quantification of lipid accumulation. This in vitro human cell model represents a valuable alternative to in vivo mice models and to primary human hepatocytes.
Thaw a nitrogen cryopreserved low-passage batch of HepaRG cells by immersing them in a 37 degrees Celsius bath until defrosted. Rapidly transfer the cells into a 15 milliliter tube containing 10 milliliters of proliferating medium. After centrifuging for 5 minutes, discard the supernatant and resuspend the cells in 5 milliliters of proliferating medium. Count the cells and dilute in order to plate 250,000 cells per square centimeter. Plate the cells on 100 mm cell culture dishes and renew the medium every 2 or 3 days. Cells proliferate with a doubling time of around 24 hours. Once the HepaRG cells reach 80% confluency, detach them through a 3 to 5 minute incubation with 0.05% trypsin solution. Collect the cells in proliferating medium and centrifuge for 5 minutes as before. Discard the supernatant and resuspend the cells with 5 milliliters of proliferating medium. Again, count the cells and dilute in order to plate 250,000 cells per square centimeter. At this stage, amplify the cells by repeating these steps to reach an appropriate number of cells to start experiments. To cryopreserve HepaRG cells, detach the cells 24 hours after plating.